discuss a side chain. Depending on the kind of amino acid, it is the side chains that differ. The structure of the side chains differ between or among amino acids. In other words, class, the amino acids will be different from each other by the presence of this side chain. But the primary component of amino acid is the carboxyl group and the amino group. This carboxyl group and the amino group is very important in the formation of polypeptide chain. It is very important in the formation of polypeptide chain because this is where it will connect the other amino acids through their carboxyl group or through their amino group. Of course, if the side chain is carboxyl group, that means it is negatively charged, it will attach to an amino group of another amino. The amino group, the amide, which is H3N, to another amino acid. Remember this simple structure of an amino acid. There are 20 common alpha amino acids. They have carboxyl group, amino group bonded to the same carbon atom. They have different side chains or R groups, which vary in structure, size, and electric charge, which influence the solubility of amino acids in water. Assign 3 letters abbreviations and 1 letter symbols. Example, glycine. Okay, the symbol is GLY, or the abbreviations is GLY, and the symbol is G. Alanine, the abbreviation is ALA, ALA, and the symbol is only 1 letter, is A. Classifications, there are several classifications of an amino acid. These are the nonpolar type and the aliphatic R groups. The nonpolar and the aliphatic R groups. And among them are alanine, valine, leucine, isoleucine, methionine, and proline. Whereas the aromatic R groups, they are the phenylalanine, tyrosine, and tryptophan. The polar or uncharged R groups, serine, threonine, cysteine, asparagine, and glutamine. And those that are positively charged, which is basic R groups, they are lysine, arginine, and histidine. And those that are negatively charged acidic R groups, they are aspartate and glutamate. Aspartate and glutamate. Okay. So these are the classifications of amino acid based on their properties, nonpolar, aliphatic, aromatic, polar, uncharged, positively charged, and negatively charged R groups. When you are talking of R group class, these are the side chains. The polypeptides, this is the polymer of amino acid. When you are talking of the peptides or polymers of amino acid, you call that polypeptide. That is a chain or made up of several amino acids. Two amino acids can be covalently joined through substituted amide linkage, termed as peptide bond, to yield a dipeptide. In other words, if there are two amino acids that are linked or joined together, you call that a dipeptide. So the peptide bonds or the peptides that differ from the number of amino acids. The formation of polypeptides differ from the number of amino acids. So if there are two amino acids that are linked together by a covalent bond, you call that dipeptide. If there are three, you call that tripeptide. 4, 5, penta, 6, hexa. Okay, but there is also what is known as oligopeptide. When you are talking of oligopeptide, usually the number of amino acids that are linked together it is either 8 or 11 amino acids. It is a short polypeptide chain. And you call that oligopeptide. The linkage between two amino acids is formed by the removal of elements of water, dehydration, from the alpha carboxyl group of one amino acid and the alpha amino group of another. What do you mean by this? In other words, when two amino acids are linked together, there is a chemical reaction that occurs wherein water is removed, and which results to dehydration, and the removal occur from the alpha carboxyl group of one amino acid and the alpha amino group of another amino group of another. And so one hydrogen and there are 3 hydrogens that are removed, okay, causing the formation of water. That is, every time a linkage is formed, there is a removal of water. Okay. And this is usually what happened. Okay? These are 2 amino acids. These are 2 amino acids. And from here, when these 2 amino acids are linked together, water is removed. Okay? Water is removed. Okay. And this is what happened. this the so this is now where the reaction occur. This is where the reaction occur. Okay. So in this case, what happened? From these two amino acids, you look at the slide, the red one, the hydroxyl group, and the hydrogen. When these react, or there is linkage, when these two amino acids are joined, water is removed, and this is how it is formed now. Okay? And this is how it is formed. So this is now carbon. This is the carbon. You look at this one. This one is this one. This one is this one. And this one is this one. Okay? And so that is how the peptide bond, how the peptide is formed, how the dipeptide is formed by removal of water. Okay? So this is now a dipeptide bond. This is now a dipeptide bond. This is the formation of peptide bond by condensation. Okay? When two amino acids condense, a peptide bond is formed between the two amino acids. Okay. A peptide bond. So this is now a peptide bond that is formed between two amino acids. Conjugated proteins, proteins that contains permanent associated chemical components in addition to amino acids. The non-amino part of a conjugated protein is usually called prosthetic group. Types of conjugated proteins, you will encounter this. Lipoproteins, which contain lipids, glycoproteins, which contain sugar, metalloproteins, which contain specific metal ions. And so you call this conjugated proteins. And these conjugated proteins, they are usually found in the plasma membrane. They are usually found in the plasma membrane. And you know, these conjugated proteins, they are very important in the synthesis of plasma membrane. And this, like, for example, these glycoproteins, you know, these proteins in the plasma membranes, they serve as channels. These conjugated proteins, these serve as channel or gate in the plasma membrane for the passage of micromolecules such as ions, gas, and other elements. These are just a short background about proteins, about amino acid. But we know that proteins, enzymes, there are several kinds of proteins. There are several types of proteins. And one kind of protein that is very important in metabolism is enzyme. Anyone who is familiar with enzyme class? Anyone which is familiar with enzyme? Michaela, are you present?
Yes, Doc.
Can you give me an example of an enzyme?
Amylase, Doc.
Amylase. Enzyme amylase. And where can you find enzyme amylase?
In the mouth, po.
In the mouth. What particular part of the mouth? Of the oral cavity?
Saliva po, Doc.
What particular organ which release the enzyme?
Salivary glands po.
Salivary gland. Okay. So you know the function of amylase. What is the function of amylase?
Breaks down carbohydrates, Doc.
Carbohydrates, like for example, what particular carbohydrates it breaks down? What particular carbohydrate?
Starch po.
Starch. Okay. Starch. Jaira, are you present? Jaira?
Yes po, Doc.
Can you give me, Jaira, can you give me an example of the enzyme that is involved in DNA replication? Jaira? Jaira.
No? Yes. Doc.
Reaction made by the enzyme. Okay, you have to understand this very well. How the enzyme react on the substrate. This is just a classic example, especially if you are taking up organic chemistry. or general chemistry, you will see this reaction always. E stand for substrate, and then A stand for enzyme, S stand for substrate, and P stand for product. In order that the product will be synthesized, the enzyme and the substrate must bind together. It will form a complex. There should be a complex or complexation between the enzyme and the substrate. And this will bring about the formation or synthesis of a product. Okay? After that the product is synthesized, the enzyme and the product will be released. Okay? It will separate. So always think of this, that the enzyme react with the substrate, and at the end, a product is formed or synthesized. Okay. This is the reaction. Any enzyme. We know always that enzymes are catalysts. Jerwell, are you present, Jerwell? Jerwell, are you present?
Yes, sir.
What do you understand, Jerwell? What do you understand by catalyst?
Okay, it speeds up the reaction.
It
Speeds up the reaction.
is
The
a
chemical reaction.
substance. It is a substance that speeds up the chemical reaction. So
Yes,
it is
sir.
a catalyst. So an enzyme is a catalyst because it speeds up the reaction. In reactions such as substrate will be converted to product. Okay? And this is a reaction that there is a changes that occur. This is a diagram that will show that there is a changes that occur during the reaction. Okay. Energy in the biological system is described as free energy. Raquel, are you present, Raquel? Raquel, are you present?
Doc, Ma'am Raquel is having troubles connecting daw po. She will try to connect once she gets home.
She gets home.
Yes, Doc. She's currently on duty, Doc. And her
On
phone
duty?
is... Yes, Doc.
Tapos na ang duty niya, baka tapos na ang klase natin. Pag nakauwi na siya, ang ibig niya sabihin, ayusin niyang connection. Earl, are you present, Earl?
Yes, Doc.
Okay. Earl, alam mo ba itong free energy na ito sa mga biological system?
Examples po, Doc.
Yes.
Um, um,
What is that?
I don't
Very
have-
important.
I don't have idea po, Doc.
You don't have any idea what is this important energy that is involved in any chemical reaction in the biological system. Wala ka bang idea ever since naging medtech ka, Raquel? Earl.
Yes po.
Earl,
Yes po.
Thorsten, do you know this energy? This is very important in the function of all biological system.
Mm. Ang ATP, Doc?
Eh, bakit ang tagal mo, Earl? Ha? Bakit ang tagal mo,
Sorry
Earl?
po.
Ha? Talagang nag-isip ka talaga? Talagang pinag-isipan mo ng gusto you. It took you about 1 hour to remember ATP. You can just imagine that.
Hindi naman po.
What is an ATP, Earl? What is an ATP?
ATP, Doc, is adenosine triphosphate.
Adenosine triphosphate. Why is it triphosphate? Why is it triphosphate?
It has 3 phosphates, Doc, in its chemical
Structure.
chemical structure.
It has 3 phosphate group. It is called triphosphate because it has 3 phosphate groups in its chemical structure.
Yes po.
Okay. Now there is a plot. If you want to know whether enzyme is really very important in hastening or speeding up the reaction, you could make a plot of the enzyme reaction. Okay, like for example, this case. Okay, as you can see, this is a reaction, as you can see. Okay, this is the ground state, meaning that it is zero reaction. But if you're going to add the enzyme, you can see the reaction, it increase. Okay, there is an increase in the reaction. Okay, and then as the product is formed, the reaction is slowed down. Okay, until product is formed, the reaction is slowed down. Okay. So that means when the product is formed, that means that the enzyme is not anymore functioning. Just imagine, class. Okay? You just imagine when the enzyme react on the substrate, there will be a reaction. So, napakataas ng energy yan. Okay. And then, once the product is formed, what will happen? There is a decline in the energy level. There is a decline in the energy level. Okay? As you can see here, that the reaction slowly go down when the product is formed. Okay. If we are going to compare this is a catalyzed reaction, and this is uncatalyzed reaction. Okay. Uncatalyzed reaction. You can see that the reaction is in uncatalyzed, meaning there's no enzyme involved. The reaction is very low. Okay? Whereas if it is catalyzed, you can see the reaction is very high. There is a very high reaction. And then the product, there is a going down in the reaction as a result of the formation of the product. Okay, class. We will be talking of inhibition because this is very important. This is very important. We know in any biological system, especially when it involves a reaction, it always involves, there is always a possibility that there will be inhibition. Okay, inhibition. Yolimil, are you present, Yolimil? Yolimil, are you present? Okay. Remember, inhibitor competes with the substrates for the active site, meaning that it is in the active site of the enzyme. Normally, the substrate binds. Unfortunately, it is not only the substrate that could bind on the active site, but also the inhibitor. Okay. So, because of that, there is a possibility that the inhibitor and the substrate will compete on the binding in the active site. Did you understand me? Did you understand me, class? Huh? Okay. Did you understand me? That the inhibitor and the substrate, they are the one that compete for the binding, in their binding in the active site. Okay. If the inhibitor will bind first, the substrate could not bind. And what do you think will happen? Gemela. Hi, Gemela. Naka-duty ka ba, Gemela? Gemela, are you still in? Naka-kung ka ba, Gemela? Naka-protective suit ka ba? Gem?
Yes, sir.
Naka-protective suit ka?
No po.
Pero normal ka lang, wala kang nandiyan ka sa isang tabi. Ha?
Nagpa-process po ng dugo.
Nagpa-process ka ng dugo. While you are processing ng dugo, you are listening to me.
Yes, sir.
But you are not protected. Mayroon kang face mask, face shield. Anong
Face
protection?
mask and gloves po with face
And gloves
shield.
lang.
Yes, sir.
Oo, anong ginagawa? Why do you have to process? That is for what? RT-PCR?
No, sir. For yung mga blood donors po.
Blood donors.
Yes, sir.
Okay. Blood donors, not for Not for COVID.
Yes, sir, not for COVID.
Not for COVID. Okay lang yan. That is not for COVID. Okay. Okay. Do you know Gem, if the inhibitor will be the first one that will bind into the enzyme? Do you know what will happen when the inhibitor is the first one that will bind into the enzyme? Do you know what will happen? Jem, are you still there?
Yes, sir.
Did you understand the question? Are you with somebody?
Yes,
Are you
sir.
with
Sorry po.
with someone? Okay, you could ask
Yes,
that
sir, my
someone.
workmate.
You have a workmate there? Mayroon ka bang workmate?
Yes, sir, meron po.
Katabi mo siya? Is she a male or a female? Jem,
Female po.
dapat ang kasama mo dyan male para you will be inspired. Okay, did you understand my question? What if the inhibitor is the first one that will bind in the substrate? What do you think will happen? Oh, is the first one that will bind in the enzyme. What will happen? Jem,
Yes, sir.
yung kasama mo dyan, is she listening to my question?
No, sir, she's talking to someone po.
She's talking to someone. Okay. Okay. Iber, are you in? Iber? Joyce?
Yes, Doc.
Okay, did you get the question? What will happen
Yes, Doc.
if the inhibitor will first bind into the enzyme, then the substrate?
If the enzyme binds first, it can stop a substrate from entering the enzyme's active site, Doc.
Huh? What again?
It can stop a substrate from entering the enzyme's active site, Doc.
So what will happen? What will happen if that will happen? If that thing will happen? If that thing will happen where the substrate is not allowed to bind into the active site of an enzyme, anong mangyayari ngayon kung maunaan siya ng inhibitor?
Doc, no reaction will happen.
And so, if there is no reaction that will happen, and so, what? If there is no reaction, so anong mangyari? Norisa,
Yes po, Doc.
are you listening to my question?
Yes po.
Yung last na question, what will happen if the substrate could not bind into the enzyme because of the presence of inhibitor?
Bali, the Typically, Doc, the inhibitors looks like the substrate for the specific enzyme. So yun nga po, if an inhibitor attach to the active site of the enzyme, it will prevent the substrate to attach to the original substrate.
So what will happen now if the substrate will not attach to the active site because of the presence of inhibitor? What will happen?
It will lead to informing a complex.
Of course, magkakaroon ng complex yan kasi nag-bind ang inhibitor sa enzyme. Pero anong mangyari ngayon? What will happen to the reaction? Peter, do you know the answer, Peter?
Hi, Doc.
Yes.
Doc,
Are you,
if
Yes.
the inhibitor is the first one to attach to the active site, there will be no enzyme substrate product leading to no reaction or catalysis or breakdown.
so what is the answer?
There will be no reaction, Doc, or catalysis or breakdown of the enzyme.
And if there will be no reaction or catalysis, what will happen?
There will be no enzyme substrate product, Doc.
There will be no enzyme, there will be no product that will be synthesized.
Yes, Doc.
In the enzyme substrate product. So, if the enzyme will not bind into the substrate, if the substrate will not bind into the enzyme because of the presence of inhibitors, product will not be synthesized or it will not be formed.
Yes, Doc.
Did you understand, Larissa?
Yes po, Doc.
Ivor, did you understand that?
Yes,
It's very
Doc.
simple. Kaya sinasabi ko sa inyo from the start of my lecture, you understand the enzyme substrate reaction to form product. That is the normal reaction. The product will bind into the enzyme, the substrate will bind into the enzyme, and then a product will be synthesized. That is the normal. Ngayon, kung may inhibitor ngayon na nag-bind sa enzyme, there will be no product that will be synthesized. And that is why, that is the reason, there will be no reaction. There will be no reaction. That is why, kung babalikan nyo yung mga drugs, yung mga drugs ngayon, like for example, the drugs that is used for arthritis, for uric acid. Anybody who is familiar with uric acid and what is the enzyme responsible for the synthesis of uric acid? Familiar ba kayo dyan? Yes, Modo. Yes. Who is that? Are you familiar? What the manufacturer did in order that, what is the role of that enzyme? The role of that enzyme is to break down, is to synthesize certain metabolite to form uric acid. Okay, and so what happened now? The company or the drug manufacturer developed an inhibitor that will inhibit the function of the enzyme. That will convert a certain substance to uric acid. Huh? And that enzyme, you call that glucose oxidase. Okay, that is the role of competitors. Because the inhibitors are structurally similar to the substrate. That's why they could bind into the enzyme, because the structure of the inhibitors and the substrate is similar, or they look alike. And so they could form an E1 complex, but without leading to catalysis or breakdown. Okay. So did you understand now why the inhibitors could bind into the enzyme? Because their structure is similar. The chemical composition of the inhibitors, there are somewhat similar to the substrate. Okay, and that is competitive inhibition. Okay. Salome, are
Yes
you in?
po. Yes po.
Okay, can you again explain or describe what a competitive inhibition is?
Back din po sa competitive inhibition, yung pong inhibitor ang magbabind dun sa active site po ng enzyme, magkakos po na wala pong reaction, kaya po wala pong magiging end product po.
Okay. Okay. Hindi mo ba, can you not speak in English? Kasi we have somebody who is foreigner.
Ay, sige po. Para lang po ma-explain ko po mabuti. Bali, sa competitive inhibition po, the inhibitor binds with the enzyme, causing no reaction and no product will be produced.
And that is competitive inhibition. Actually, when you are talking of competitive, that means that the inhibitor competes with the binding in the active site, competes with the substrate in the binding in the active site, on binding in the active site of the enzyme. And that is competitive inhibition. Huh? There is competition. And do you know the reason why? Maria Salome, are you present?
Yes po.
Do you know the reason why the inhibitor could bind into the substrate, into the enzyme?
The inhibitor resembles the substrate.
No, the inhibitor, why the inhibitor could bind into the substrate, into the enzyme? Why the inhibitor could bind into the enzyme? Do you know the reason why?
No idea, Doc.
No idea. I explained that just a while ago.
I'm trying to follow pa po, Doc.
Huh? I explained that. Jem.
Yes, Doc. Yes, Doc.
Ano ba ang pangalan mo, Jem?
Jem po.
Taladhay?
Yes po.
Jem talaga.
Yes po.
Okay. Did you hear my question?
Not clearly, Doc.
Not clearly.
Yes po.
You are sleeping. I repeat. Why is
Okay.
it that the inhibitor could bind into the enzyme?
Because they have similar structures. That's why the inhibitor could easily bind to the enzyme.
Maria Salome.
Yes po,
Maria
Doc.
Salome,
Yes po.
can you hear what Jem explained or answer?
Yes, Doc.
Okay, can you repeat that again?
The inhibitors resembles the structure of the substrate so that it will easily bind with the enzyme.
May ano dyan sa inyo, Jem? May ano dyan sa inyo, Maria? Ha?
Po?
May anong nangyayari dyan sa inyo? May happenings ba dyan sa inyo?
Ay, hindi po sa akin yun, Doc.
Ay, sa amin, Doc, yung kapitbahay po namin, like,
Kapitbahay?
nagko-construction po. So,
Ah,
medyo rinig po yung conversation nila.
ganun ba?
Opo,
Magpalagay ka
po.
nga, magpalagay ka nga dyan ng anong pwede mong ilang acoustic sa bahay mo para hindi mo marinig ang mga yan so that you could not hear the voice of those of your neighborhood. Okay.
I'll try, Doc, if may budget.
Maria, again, Maria. I could not understand you, Maria.
Doc, the inhibitors resembles the structure of the substrate that makes it easily binds with the enzyme.
The inhibitor resemble to the structure of the substrate. That is why it could bind into the enzyme.
Yes po.
Okay, Maria. Now we could go with uncompetitive inhibition. Okay. From competitive, then we go to uncompetitive inhibition. Okay. Which binds a site distinct from the substrate active site. And unlike competitive inhibitor, binds only on the enzyme substrate complex. Whereas an inhibitor under uncompetitive inhibition, the inhibitor binds at a site different from the substrate active site. Did you understand, class? In other words, class, the inhibitor will not bind into the active site. But there is another binding site. There is another binding site in the enzyme where the inhibitor could bind. Meaning that the enzyme, it has an active site, and besides that, it has another site. So, the inhibitor will not compete on the enzyme because they could bind simultaneously. The inhibitor and the substrate could bind simultaneously into the enzyme. Unlike in the competitive inhibitor, that the substrate could bind only on the active site. Similarly, the inhibitor could bind only on the active site. Okay, did you understand this, class? Why it is called uncompetitive inhibition? Zora, Vincent?
Yes.
Okay? Did you understand my...
Yes.
Did you understand my
I did.
explanation? Why it is uncompetitive inhibition?
Yes.
Have you explained that to Sora?
Yes.
Okay? Can you explain that again? Why it is uncompetitive inhibition?
Okay.
Can you explain it again?
Yeah, I'm explaining right now.
Yes?
Yes.
How do you explain that? No, you just explain to me. I mean, explain to me about this. Hello?
Yeah. I think the substrate have two binding bonds. Sites. Yeah.
Sites.
Okay. And so it makes the inhibition not very competitive.
Yeah, that's right. The substrate has two different active binding sites. Active binding sites. These two active binding sites could bind simultaneously the inhibitor and the substrate. Could bind simultaneously on the inhibitor and the substrate. Unlike with competitive inhibitor, wherein there is only one binding site, and that is the active site. How do uncompetitive inhibition differ from mixed inhibition? Inhibitor binds at a site distinct from the substrate active site. This is similar to uncompetitive inhibition. Okay? But it binds either to enzyme or enzyme substrate. But it binds either to enzyme or enzyme substrate. Mixed inhibition. Okay. This is how for competitive inhibition look like. Okay. Okay, so this is the normal. Okay. This is the normal process for enzyme substrate complex reaction, for enzyme substrate reaction. The enzyme binds on the substrate to form enzyme and products. But in the competitive inhibition, the substrate will usually bind into the active site. This is the active site. Okay? Similarly also, this is now the substrate. The inhibitor could bind also in the active site. It could also bind into the active site. Okay. So the enzyme now could either have the inhibitor or the substrate. But once the inhibitor binds into the active site, the substrate could not bind anymore. It could not bind anymore. So a product will not be synthesized. So a product will not be synthesized. The reason why there is competition is because it has something to do who will bind first into the active site. It is either the substrate or the inhibitor. So there will be competition who will bind first. If it is the substrate that will bind into the enzyme, a product will be formed. But if it is an inhibitor, the enzyme will be inhibited. The enzyme will not be inhibited, meaning the enzyme could not form or could not break down the substrate to form the product. Okay, class. Did you understand this diagram about competitive inhibition? Okay? Michaela.
Yes, Doc.
Michaela, did you understand my explanation?
Yes po.
Okay. Can you explain this diagram? Can you explain this diagram?
Yes, Doc. So in the diagram under the competitive inhibition, it shows that the substrate and the inhibitors are very resembles each other in structure. So due to this, both of the substrate and the inhibitors have equal chance to actually bind to the enzyme. So if the inhibitor was able to bind to the enzyme, no reaction or catalysis would occur. Therefore, there would be no product formed.
Okay. Based on what of your observation, Michaela,
Yes, po.
do the inhibitor and the substrate have the same? They have the same structure, similar. They have similar structure.
Not exactly, Doc, but they have part of the inhibitor. The inhibitor has a part that is similar to the substrate. So this
Okay,
part po, which
okay.
is
That's right, Michaela. That is only partly. Yeah, that's partly. That is the structure that could be inserted or could be bound into the enzyme. Okay. And that is competitive inhibition. Okay. That means the substrate and the inhibitor compete with one another. Compete with one another so that compete with one another in binding to the active site of the enzyme. The active site of the enzyme. Okay, here the diagram. In uncompetitive inhibition, Okay? Okay, you could see the enzyme, then the substrate, okay? The substrate, okay? The enzyme class, it has two binding site. the enzyme has two binding site. This one. Okay. First, you have the substrate. The substrate is bound into the active site. And there is another one, which is also an active site, where the inhibitor could bind. Okay? It could also bind. And so what happened? If the inhibitor simultaneously bound with the substrate, what do you think will be the reaction? Is there a reaction in air? Uncompetitive. Is there a reaction? What will happen? What do you think will happen? You look at, if both the inhibitor and the substrate bind into the enzyme, where there will be a reaction. Jaira, you present, Jaira? Jaira? Angel? Angel?
Yes, po, Doc.
Okay. What do you think is this reaction, Angel? If there is uncompetitive inhibition, if the two simultaneously bind into the enzyme, is there a reaction that will be formed?
No effect, Doc.
No effect. If there's no effect, so what will happen? If there's no effect,
If there's no effect to Doc, it makes more difficult for to convert the product po. Increasing the enzyme affinity po.
okay. In other words, product will not be formed. Okay?
Yes po, Doc.
Product will not be formed if the inhibitor simultaneously binds with the substrate in the enzyme. Always refer to the normal reaction. The normal reaction, product is synthesized. That is the normal reaction. Product is synthesized. So in mixed inhibition, okay, it is somewhat similar to uncompetitive inhibition. Okay, it is similar to competitive inhibition. The substrate will bind and the inhibitor will bind. Mix. Okay? Or simultaneous. Okay? Mix. The inhibitor or the substrate could bind, could be mixed. Okay? the substrate and the inhibitor could be mixed, and then what will happen? It will bind into the enzyme. Okay. Okay. There is a mix, but also mixed binding. Okay. And in this case also, because of the mixed inhibition, product will not be synthesized. So always remember this class, because later on you will learn that the presence of inhibitor could affect the transcription process. It could affect the translation process. So, when the translation process is affected, so the polypeptide chain will be also affected. If the translation of the messenger RNA will be affected because of the presence of inhibitor, the synthesis of the polypeptide will be also affected. And you know, when this polypeptide will be affected, it will change the structure of the protein. So what will happen? The protein or the enzyme could not bind into the substrate. Who will report on glycolysis?
Ako po, Doc. Yes, ako po.
What's your name?
Angela po.
Angel.
Yes po.
Have you already seen your report? Have you already completed your report? Glycolysis?
Not yet po, Doc. Pinag-aaral lang po na mabuti.
Nakita mo ba kung ano ba ang what is your report? Glycolysis.
Yes po, glycolysis, Doc.
Hindi ka ang first reporter. Dapat ang first reporter, the one that will report on the transport of glucose into the cell. Who will report on that?
I will be the one to report that, Doc.
Oh, okay, you'll be the one. You will be the first reporter.
Yes,
Okay.
po.
And then second will be Angel. If you
Yes,
have,
Doc.
if you have already looked at your pathway, Angel, if you have already go over the pathway, you will see that there are several enzymes that are involved in the synthesis of a particular product. Like, for example, when glucose enter the cell and glucose undergo glycolysis, the first product that will be synthesized is what, Angel? From glucose. Ha? Hindi mo pa alam? Ha?
Doc, can you repeat po? Hindi ko po na masyadong narinig.
When glucose enter the cell and then it undergo glycolysis, what is the product that is first synthesized? What is the product that is synthesized?
The product that synthesizes the glucose po, Doc,
Ano?
lalahanin ko lang, Doc.
Wala, hindi mo pa nagawa.
Ano to, Doc, sa ano, it takes in the cytoplasm muna, Doc eh. Then the glycolysis po, yung the first step is to
Ano,
break.
ang pathway, ang i-report mo dyan, yung glycolytic pathway. O glycolytic pathway ang i-report mo dyan. When glucose is converted to what?
Okay po, yung ano po, yung glucose 1-phosphate. Ayun po ba, Doc?
Glucose 1-phosphate ba?
Yung pinakauna po.
Glucose 1-phosphate?
Doon po muna sa,
Glucose
sa
1-phosphate ba? Tingnan mo, do you have your metabolic pathway?
yes po, Doc.
Anong nakasulat dyan? Glucose 1-phosphate. Can you see your pathway?
Yung ano,
May
Doc,
arrow yan eh. Glucose and then may arrow yan pababa. Nandoon yung product glucose what?
glucose 6-phosphate po, then maging glucose fructose 6-phosphate.
Glucose 6-phosphate.
Opo.
Hindi ka marunong magbasa ng pathway, Angel? You do not know?
Ibang ano po, ibang step po yung na nakita ko po, Doc.
Ibang nakita mo.
Ibang step po. Not
Ang glycolysis hindi magbabago yan. It will always be glucose 6-phosphate.
Okay po.
Ang first product of glycolysis is glucose 6-phosphate.
Opo.
Tanda mo yan. Angel, glucose 6-phosphate. And then what is the enzyme that is involved in the synthesis of glucose 6-phosphate?
The hexokinase 1 po, Doc. The hexokinase po.
Hindi ko marinig.
At napalalabas na. The enzyme that
What is the enzyme? Sa side yan, makikita mo.
yung hexokinase po, Doc, glucose to glucose 6-phosphate po, yung enzyme po na is the hexokinase po.
Hexokinase.
Yes po.
Okay. Nakita mo na yan, hexokinase. Makikita mo other enzymes, other reactions pagkatapos ng glucose 6-phosphate, glucose 1-6-phosphate,
Opo.
fructose, what?
Fructose 6-phosphate po.
6-phosphate,
Yes
then
po.
fructose.
1,6-biphosphate po.
Oh, okay. Yan, dapat memorize mo yan kung ang mga reaction yan and then the enzyme. Now, Angel, you can
Yes.
just imagine if there is an inhibitor. Okay? If there is an inhibitor that was added during the process of translation. Do you know, Angel, the three components, the three RNAs that are involved in the translation process?
The 3 RNA po.
Yes, the three RNAs involved in the translation process.
The 3 RNA involved in translation process are the messenger RNA, tama po ba? The
Do you have
mRNA,
your assignment? Assignment
yes,
po ba yan? Eh baka sasabihin mo kung tama, if
Ah,
that is your
the-
assignment.
wala lang
Correct
po.
kasi kinopya, you copy that from the book.
Okay po. The mRNA, or what we call the messenger RNA, the rRNA or ribosomal RNA, and the tRNA or what we call the transfer RNA po.
Okay. Just imagine if there is competitive inhibition, okay? If there is uncompetitive, if there is an inhibitor, just imagine if your ribosomal RNA is inhibited. Okay?
Opo.
If an inhibitor binds into your ribosomal RNA, what will happen? What will happen, Angel, if an inhibitor will binds into your ribosomal RNA?
If the inhibitor po binds to the ribosomal RNA, it may be po it diminishes the rate of catalysis po by reducing the proportion of enzyme molecule.
Walang enzyme dyan. Ang ribosomal RNA ba is an enzyme?
Hindi po. I mean po, pag magkakaroon po ng ano,
What will be affected if an inhibitor will bind into the ribosomal RNA? What will be affected?
yung maapektuhan po yung
You do not know the function of the ribosomal RNA.
Dok, pa ulit po ulit, Dok. Iniisip ko po masyado.
Iniisip mo ha? Isipin mo muna ha? Somebody could answer that. What will happen if the inhibitor binds into the ribosomal RNA? Anybody from the class? Anybody from the class? What do you think will be affected if the inhibitor will bind into the ribosomal RNA? Pax?
Doc,
Yes.
Since the RNA is inhibited, I think the translation will not occur.
Yes, that is right. Para kang natatakot, Pax, sumagot. Huh? Parang you are afraid of answering, Pax. That is correct. The translation process will be affected if the inhibitor. And so, if the translation process will be affected, Pax, what will happen? Anong mangyayari if the translation process will be affected?
I'm sorry, Doc. What will happen if
the translation process will be affected because of the presence of inhibitor in the ribosomal RNA, what will happen? What do you think will happen if the translation process is affected?
there will be no new DNAs.
Bakit no DNA? Translation yan. That is translation. Why DNA?
Since translation is affected, so since it's part of the process of translation, then when there is an inhibition, and eventually it affects the whole process, so there will be no product produced.
And what is that product?
The RNA.
RNA ba ang product? Paano RNA ang product? Translation, RNA ang product? That is right. So the translation is affected. So when the translation is affected, what will happen? The synthesis of polypeptide will also be affected. There will be no synthesis of the polypeptide because the reason why is the messenger RNA could not bind into the ribosomal RNA because of the presence of inhibitor. And if the messenger RNA could not bind, so translation will not occur. If translation will not occur, polypeptide will not be synthesized. Huh? Alam nyo, kailangan yan, your mind should always focus on what is going on. Huh? Always what, why, and how it will happen. There should be three things that should happen into your mind. The question of how, the question of why, and the question of what. Okay? Okay. So you know that the enzyme is affected by several factors. One is the pH. The enzymes only acts or functions. Enzymes functions at an optimum pH. If it is higher or lower pH, the enzyme activity decreases. Okay. That is why when you are working with enzyme, you should always have the right pH, and the right pH, it should always be normal or a pH range between 6.8 to 7.2 in order that the activity of the enzyme will not be affected. Okay, this is what happened. Okay, this is the pH optimum. Okay, the activity of the enzyme increase when the pH is at 7, and then it levels off, and then the activity of the enzyme goes down as the pH level increase or become acidic. So this is the optimum activity of the enzyme. It should be pH 7. it should be at pH 7. Okay? You look at this one, there is an increase at pH 7, at pH 7, okay, the activity of the enzyme is stationary, and then as you increase it further to 8, there is a decline in the activity of the enzyme. There is a decline in the activity of the enzyme. Okay. Regulatory enzymes, allosteric enzymes, function to reversible non-covalent binding of regulatory compounds called allosteric modulators or allosteric effectors, which are generally small metabolites or cofactors. Effectors, the cofactors, small metabolites, multi-subunit proteins, and some cases the regulatory site and the active site are separate units. Some enzymes are either stimulated or inhibited when they are bound by a separate regulatory proteins. So there are allosteric modulators or allosteric effectors. That is why what are these cofactors? You know what are these cofactors, class? Small metabolites? The allosteric enzymes have different structures from non-regulatory enzymes. Allosteric enzymes have one or more regulatory or allosteric sites for binding the modulator. Regulatory site is specific for binding of modulator, generally larger and more complex than non-allosteric enzymes, and 2 or more subunits. We are talking here of allosteric enzymes. So, the regulatory sites of the allosteric site is specific for binding of the modulator. Okay, like for example, this one. This is the substrate. Usually this is the regulators and the catalyst. Okay, this is the enzyme. the substrate binds, the modulator binds, what happened to the enzyme? The enzyme becomes more active. And this is the active enzyme substrate complex. This is the regulator, this is the substrate. Here you can see that the enzyme needs a positive modulator in order that it will become more active. Okay. The enzyme, the regulator, the enzyme, the regulator. This the substrate binds into the enzyme, and then the modulator binds into the regulator, and then what will happen? The enzyme becomes more active. This is an active enzyme substrate complex. Okay. Okay, class, I think we have to stop in here. Okay, do you have any question, class? So next meeting will be our long exam. So I'm going to give you the questions. Next meeting, I'm going to give you the questions, and then the following morning, you have to submit your answer. You have to upload your answer in the Google Classroom. And also the first reporter also, the first, the second, the third, the fourth, and the fifth will also report next meeting. Who is the first reporter?
I'm the first reporter, Doc.
Michaela?
Yes po.
Okay. What is your report, Michaela?
Glucose transport po into the cell.
Okay. Then Angel, glycolysis.
Glycolysis po, Doc.
Jaira is absent. Jem.
Yes po.
Are you check if this Jaira still will be also attending our class? If Jaira?
Yes, sir.
Paki-check mo lang. Then Alexander.
Yes,
Then
Doc. Hello.
Abdul. Okay. You will be the first reporter. And do not be absent. And then also, Pax, you try to prepare now your report. At saka, and then also the coverage of our exam will be the first, the transport of protein, the mechanism of transporting proteins, then the structure of the DNA, then transcription, translation of the DNA, and then last will be the repair mechanism. So I'm going to give you the question before we start before we start the reporting, I'm going to give you the questions of the long exam. And then the following morning, you have to upload your answer to the Google Classroom. Okay, do you have any question, class? May mga tanong ba kayo?
Sir, yung... Doc,
Yes, Angel.
Doc, yung exam po namin, kinabukasan po yung submission.
Yes,
Okay.
you have to submit that before 8 o'clock. You have to upload your answers to the Google Classroom. I want to see that.
Noted po, Doc.
Huh?
Noted po.
Kaya you have to prepare all your, lalo na yung assignments nyo. Pagbutihin yung pagsulat dyan sa mga assignments nyo. Huh? You should always have a good written assignment. Abdul? Okay?
Yes, Doc.
That assignment will help you a lot during the exam. Okay. So, see you again next meeting. Jim, what day is that? October what?
October 2 po.
October 2. Okay.